In this video, we're going to talk about the structure of DNA, in that structure of nucleotide. DNA. The DNA stands for the deoxyribose nucleic acid. Deoxyribose nucleic acid. So it is a type of nucleic acid. It is a type of nucleic acid. And it was for the first time reported by Frederick Miescher. The DNA was first time reported by Frederick Miescher, and the term DNA given by Jackarich. The term DNA was given by Jackarich. Jackaris or Jackaris, whatever. And the structure of DNA, was given by Watson and Crick. This structure, which was explained by Watson and Crick, is double helical model. This is nothing but double helical model. And this demonstration of structure of DNA by Watson and Crick, it was based on the data that they have collected from the Wilkin and Franklins. Okay, Wilkin and Franklins. They have conducted the X-ray diffraction method. So Wilkin and Franklin have conducted X-ray diffraction to understand the structure of DNA. So based on the data, this Watson and Crick, they have demonstrated double helical model, Watson and Crick. So this was just a small historical view of the DNA. Okay. Now, what is DNA? Nucleic acid. The name itself we can understand. This is an acid present in the nucleus. Nucleic. See, we are Indians because we are living in the India. It is nucleic because it is present in the nucleus. So, and basically it is acid. It is an acid. And it is present in the nucleus. So, nucleic acid. So, this was just a small introduction about the DNA. Now, we are going to study about the structure of nucleotide in this video. Nucleotide. Deoxyribose nucleic acid, in short, DNA, is a polymer of nucleotides. The DNA is a polymer of nucleotides. It's a polymer of nucleotides. Why? Because there are many nucleotides joined to each other which forms the DNA. And coming to nucleotides, these nucleotides consisting of three components. There are three components in this nucleotides. The first one, phosphate group, and second one, pentose sugar, and third one, nitrogen base. So totally three components will form the nucleotide. There are three components. These three components will form the nucleotide. First group is phosphate group. Next pentose sugar, next nitrogen base. So how they are present, I'll explain now. First pentose sugar will start with. So this pentose sugar is nothing but deoxyribose sugar. deoxyribose sugar. And this consisting of 5 carbons. Why? Because it is 5 carbon. Penta means 5. This pento. Penta means 5. Os. OS is a suffix of sugar, right? And this is the first carbon and second carbon, third carbon and fourth carbon. And the fifth carbon will be present as an arm. The fifth carbon will be present as arm. And coming to nitrogenous base, the nitrogenous base will be attached to, the nitrogenous base is attached to the first carbon of the pentose sugar. The nitrogenous base is attached to the first carbon of the pentose sugar. And this bond is called, and this linkage is called N-glycosidic linkage. N-glycosidic linkage. N-glycosidic linkage. Glyco means sugar. Glyco means sugar. So N for nitrogen and glyco means sugar, this pentose sugar. So N-glycosidic linkage. Nitrogen base is attached to the first carbon of this pentose sugar, deoxyribose. And we may get a question, so what about phosphate group? The phosphate group is attached to the fifth carbon. The phosphate group is attached to fifth carbon of this deoxyribose, and this linkage, this bond is called phosphodiester bond. Phosphodiester bond. Phosphodiester bond. So totally all these 3 components, number 1 phosphate group, number 2 pentose sugar which is deoxyribose, and number 3 nitrogenous base. And this nitrogenous base is attached to the first carbon of the deoxyribose, that is called N-glycosidic linkage, and the phosphate group is attached to fifth carbon of the pentose sugar by phosphodiester bond. And coming to nitrogenous bases, there are 2 types of nitrogenous bases. There are 2 types of nitrogenous bases. So number 1, purines, and number 2, pyrimidines. Number 2, pyrimidines. So purines are again 2 types, adenine and guanine. Adenine and guanine. And pyrimidines are 3 types, cytosine, and thymine and uracil. So these are the nitrogenous bases, purines, adenine, guanine, and pyrimidines, cytosine, thymine, uracil. 3 types of pyrimidines and 2 types of purines. And this nitrogenous base is attached to the pentose sugar, to the first carbon always, to the first carbon. It is the first carbon. Coming to, this is deoxyribose, right? And here functional groups are present. Hydrogen, OH group, OH functional group in this deoxyribose. So this is a structure of nucleotide. So hope you understood.